Hello everyone and welcome to Aloasis Academia. This is a new session, and in this session we will learn about methods of studying cells, specifically microscopes. Okay, so the first tool we use to study cells is microscopy. So what's a microscope? Microscope is a tool that makes very small objects look bigger so we can see them. And in this context in specific, when we say an object, we refer here to the material that we put under the microscope we are looking at it. So that's an object, that the thing that we are looking at through the microscope. Why do we need a microscope? First of all, because many biological structures are too small for the human eye to see them. It's very important to know that the human eye can see objects only down to 0.1 millimeters. So anything that is smaller than 0.1 millimeters, you will not be able to see it with your naked eye. And that's why you would need a microscope. Here are a few examples to have an understanding of how different cells would have different sizes. So the first one is a human egg cell. It's about 1.1 millimeters, so it's just visible to the naked to the eye, and it is very clear under the microscope, as we can see. However, when it comes to the red blood cells, they are 7 micrometers, so they are quite small. And as we can see under the microscope, we see very small circles, and we cannot see anything inside them. So they are not very, very visible. When it comes to bacteria, which is around 1 to 2 micrometers, it's completely invisible. We cannot see anything inside the cells. We cannot identify the borders of each bacterial cell, so it's absolutely invisible. And that's why we would need a microscope to be able to see these things and study them. So, now we need to understand, how does a microscope magnify things? So the way the microscope would need to make things bigger is by using lens. So a lens would bend the light that is passing through the lens, and this would make any object looks bigger. An example of that is the magnifying glass, which has a simple lens in it, and this lens will just make all the letter appear larger. To understand the concept in a better way, as we can see here, so we have this original object here. So the light will be reflected. If you are in a lit room, the light will be reflected from the object through the lens. Once it passes the lens, it will bend, and then the human eye will try to trace the light backward in straight lines, and that what makes the human eye see the object in a larger way than it is in the original form. And that's how basically microscope would work. So what we just explained so far is how the simple microscope would work, which is like the magnifying glass. However, when it comes to the light microscope we are using in science and in the labs, it's a bit different because it's even stronger in terms of making the object looks even bigger. And this light microscope is called the compound light microscope, and it uses two lenses. One lens gonna be the ocular lens, which lies here, and then you would have another lens called the objective lens. And then the object will be placed on the stage there. So whatever cells you have, whatever tissues you have, you will place them on the stage there, and then you will look through this side, this position here. So your side, you will look through the first lens, which is ocular lens, and then look through the objective lens, and then you see the object. Because they are using two lenses here in the light microscope, this what makes the magnification even higher, and you would see the object even higher. And to explain that, each lens here, the ocular lens or eyepiece lens and the objective lens, they magnify object on their own. So for example here, the eyepiece would make the object looks bigger, 10 times bigger, and the objective piece will make the object looks 40 times bigger. If that's for each one on its own. So if you look through the microscope in this position, you will go through the eyepiece lens and the objective lens. So the total magnification you would have is the product of both of these lenses. So the eyepiece, which makes things bigger 10 times bigger, and then the objective lens, which makes the object 40 times bigger. So in total, you will see the object 400 times bigger than it is in real life. And that's why it's very useful to use compound light microscopes to study cells and tissues, because you would be able to see them very clearly and be able to study them in the lab. So to improve your understanding of microscope and how microscopes work, you need to understand what does magnification mean, because magnification is a big part of how the microscope work, and that's why it's actually the main function of the microscope. So when we say magnification value, it just tells us how many times bigger the image is compared to the real object. That's what magnification is. So when we say a magnification of 400, it means that it's a magnification that makes the real object 400 times bigger. That's what it means. So you need to understand that there is always a difference between the object size and the image, or what you look under the microscope. So the object, as we said, is the real thing that you put on the stage on the microscope and you look at under the microscope. The image is what you see through the microscope or in an image. So why there is a difference? As we said before, because the microscope or the compound light microscope, they use two lenses that enlarge the object, and the total magnification is the product of the enlargement of each of these lenses. One important formula that you need to learn by heart is the formula of magnification. So sometimes there's a lot of exam questions that comes on that asking you to find the magnification value for something in a question. So to find the magnification, what you need to do is basically dividing the size of the image by the size of the real object. So what we are saying actually is how big it looks divided by how big it really is, and this will give you how many times the real object has been magnified. In exams, it's very common that they might ask you questions that requires you to rearrange this equation, the magnification equation formula. So what we have, sometimes they might ask you to find the real size of an object, and in the question they will give you the size of the image and the magnification value. And in that case, you just rearrange the equation. So the real size of the object is the size of the image divided by the magnification. One of the very common traps that students fall for is that they do not make sure that the units are the same for the size of the image and the size of the object. Always make sure that both are on the same unit. So if one value is in millimeters, so for example, if they have given you that the image size is in millimeters, you need to turn the real object size into millimeters as well. Okay, otherwise the whole calculation will be incorrect. It's like dividing meters by centimeters, so everything will be incorrect, and you will lose a lot of marks for something that easy. Always remember that you need to rearrange the equation in the right way and make sure that the units are correct and the same. So now I will give you a quick example to show you or to prepare you for the questions that can come up in the exam. So for example, in the question of the exam, they will tell you that, for example, a real size object has a size of 100 nanometers and an image size is 10 millimeters. And what they need you to find is the magnification. So how many times does the real object needed to be magnified to be able to see it in an image size of 10 millimeters? And it's an easy process. If you just take it step by step, you'll find it a very easy way. So the first thing you need to do is to convert everything to the same unit, because as you can see here, image size is in millimeters. However, real size is in nanometers. So these are two different units. My recommendation is that you always try to when you want to make the units the same, always try to make them to convert the bigger units into the smaller units, because it's just easier. You just multiply by something. So for example, in this case, if you convert the millimeters to nanometers to make everything in nanometers, all you need to do is that you times 10 millimeters by 10 to the 6 by a million, and this will give you 10 million nanometers. So that's the first step. Once you converted all the units all the values to the same units, then you just use the formula and plug in all the values. So the size of the image you don't plug in the 10 millimeters, you plug in the 10 million nanometers, because we have converted it now by times it by 10 to the 6, and then divide it by the real size of the object, which is 100 nanometer, and then this give you 100,000, which means that the magnification is 100,000. What does that mean? It just means that the real size, the real object is really small. It's just 100 nanometers, and it required us to magnify it 100,000 times to be able to see it in an image, which is 10 millimeters. And that's the whole thing. So always remember that there are two steps. Always split your work into two steps. First one, make sure that the units are the same. The second one, just apply the formula and make sure that you are arranging the formula in the right way. And in this question and this example here, they are asking for the magnification, so no rearrangement was required. So here we have quick reminders. So it's very important that you learn the conversions between the units, because that will make your life much easier, and it will save you a lot of time during the exam. So in this table it shows you if you're converting from kilometers to meters, you just times by 10 to the 3. From meters to meters, time to the 0, which means just 1. It's the same thing because it's the same unit, meter. If you convert it from millimeters to meters, which is from the smaller unit to the larger unit, you divide by a 1,000. However, if you are doing it in the opposite way, from meters to millimeters, you will times it by 1,000. And the same thing with micrometers. If you are converting from micrometers to meters, you divide by 1,000,000. If you are converting from meters, which is the bigger, to the micrometers, which is the smaller, you just times it by 1,000,000. And when it comes to the magnification triangle here, it's just an easy way to remember the formula. So magnification will equal image size over the real size. Image size is magnification times real size. Real size is magnification is the image size over the magnification. It's very important that you learn these two things, as this will save you a lot of time during the exam. Okay, so the second most important thing about microscopes, it's a very important concept after magnifications. It's called resolution. So as we said, magnification is making the real object looks bigger in the image, or even under the microscope, when you are looking at through the eyepiece, you will see the object in a much bigger way. Resolution, it's different. So resolution, sometimes you will find it referred to as the resolving power, and it's the minimum distance between two objects so they can be seen as separate. So what does that mean? So if you are looking through the eyepiece of the microscope and you're looking at an object, what resolution is, if you are looking at 2 points on the objects, if the 2 points are far enough apart, these 2 points will be seen as two separate points. If they are too close together, then they will appear as one blurred point. And that minimum distance is called resolution. So for an example, if you are looking at this captured image here, a low resolution image would give you something like that. So you cannot see the details of the image, and most of the points, or the vast majority of the points, will be just shown as blurred points. So that's a low resolution image. For a high resolution image, it's a sharp image where you can see the details of the image you have. So if you pick any 2 points on this image, you can see them clearly, which means that they are far enough apart to see them as two separate points. However, in this image the 2 points are too close together, so they appear as one blurred point. That's why the whole image would look blurred. So what does affect resolution, or what resolution depends on? So what makes an image or a microscope gives you a low resolution image, and another microscope might give you a higher resolution image, that all depends on the wavelength of the radiation that it's used in the microscope. So in light microscopes, the one that we just covered, we are using, obviously, as the name hints, light waves. And light waves has relatively long wavelength. And when it comes to microscopes, long wavelength will it mean just low resolution. So if you have a wavelength of radiation that's even longer than light, then you will have even lower resolution. So the longer the wavelength, the lower the resolution. However, when it comes to other type of microscopes, which we will cover in different videos, so electron microscopes, for example, they use electrons which have shorter wavelength. So shorter wavelength, which means higher resolution. So just for a hypothetical example, if we have these two images here, so this one will be taken by a light microscope because it has a low resolution. An electron microscope will show you a much sharper image, which means that you will see more details in the image. Okay, so that's the main difference. It depends on the wavelength that is used, the wavelength of the radiation that is used in the microscope. When it comes to light microscope, the resolution is 0.2 micrometer, and that means any 2 points that are closer to one another, or the distance between both of them is smaller than 0.2 micrometers, you will see them as just one blurred points. Okay, that's a very important point. That's very important information. It might be examined on the paper, or just maybe asked in some practical exams. So just understand that for light microscope, the limit is 0.2 micrometers. Any 2 points that are far apart enough, and the distance is 0.2 micrometers, they can be seen as two separate points. Otherwise, they will not. And here's an example. If two bacteria are, for example, 0.3 micrometer apart, so you will see them as two separate bacteria. However, if they are too close to one another, which is 0.1 micrometer, then under the light microscope, you will just see one blurred shape. You wouldn't be able to tell if if they are two bacteria or if it is just one bacteria. So this limit, 0.2 micrometer, is very important to remember. So now let's compare magnification to resolution. So hopefully this would make the whole thing much clearer to you. So if you increase the magnification of a microscope, that would make the image bigger, but doesn't always increase the resolution. Why not? And the reason for that is because every type of microscopes has a specific resolution limit. As we said in light microscope, the resolution limit is 0.2 micrometers. So if you have two bacteria that are 0.1 micrometers apart, even if you increase the magnification while you're looking at the bacteria just to make it bigger, it will not make it clearer. They will both just still be shown as a single blurred point. And that's why, as we said, increasing magnification beyond this point will just give you an image that is bigger but blurrer. Okay, so magnification and resolution are not the same thing. You need to understand this concept. So the key take-home points. Microscopes allows us to study cells that are too small to be seen. Magnification is telling us how much bigger something is than a real object. Resolution is more important than magnification because it allows us to determine the details of the image. Light microscope has a resolution limit of 0.2 micrometers. Electron microscopes use shorter wavelengths, therefore they have higher resolution and more details in the image. To complete your revision, you would need to look at the revision booklet that I have on my store on PayHip. They are sold at a very low price, but they will give you a full keyword bank for this topic. So you're gonna ace your exam just writing these keywords. Don't forget to like, comment, and turn on the notification button so you will be notified of our next topics. Thank you.